the main aim of this video is to read each and every line of the first aid and to explain the first aid book. So currently we are starting with biochemistry. Let's go step by step, don't be in a hurry. We will try to finish it as soon as possible. Okay, coming to this book, the first chapter that is given is biochemistry. So we are gonna starting up with biochemistry. First we will gonna read all these lines and I will gonna explain this. So in biochemistry, the chromatin structure. DNA exists in a condensed form. Chromatin forms to fit into nucleus. DNA loops twice around the histone octamer to form nucleosome. Beads on string. H1 binds to nucleosome and to linker DNA, thereby stabilizing the chromatin fiber. DNA has negative charge formed from phosphate groups. Histone are large and have positive charge from lysine and arginine. In mitosis, DNA condenses to form chromosomes. DNA and histone synthesis occurs during S phase. Mitochondria have their own DNA, which is circular and does not utilize histones. So here is the diagram given. So I am going to explain. DNA exists in condensed form. What they are willing to say is, see, here DNA existing in double helix form. If the DNA exists like this, your cell cannot accumulate this long DNA. So what our cell does is, it's going to condense this DNA, which is obviously double helical structure. So see, so to condense you need a core. This core is formed by histone molecules. These histones, what they do is they form the core. So around this, all this DNA revolves around this histone core. And this is the histone linker molecule. What do you mean by linker molecule? See, if there is, let's assume a cricket ball or any baseball around which a thread has been rolled around it. So when you pull out the thread, the ball revolves and the thread comes out. So to not let it happen, we are going to make here a stitch, which is nothing but your histone, H1 protein. So your histone octamer contains 8 histones. These are the histones and each 2 in number, which makes 8. So your linker DNA is this one. This is H1. So your DNA gonna loop around each and every histone octamer, forming into condensed form. See, this is the condensed form. See, this is the supercoiled structure. See, this each and every line given is DNA. See how much DNA is accommodated. This is heterochromatin. What do you mean by heterochromatin? Let's see in further lines. And the important points to take over here is DNA loops twice around each histone octamer to form nucleosome. And see, linker DNA stabilizers. DNA has negative charge. This is important because negative positive attract. See, histone has positive charge whereas your DNA has negative charge. Once these two attract each other, which make them more stable. So this negative charge is by phosphate groups, and this positive charge is formed by lysine and arginine. See, here it is given lysine and arginine, mnemonic excellent, mnemonic large. In mitosis condensed to form chromosomes, we know DNA histone synthesis happens in S phase. Yes, mitochondria have their own DNA, which is circular. This is important. Circular DNA does not utilize histones, so no histones in mitochondria. So before going to your heterochromatin and euchromatin, we are gonna go with methylation and acetylation, because if we understand methylation and acetylation, then we can easily understand the euchromatin and heterochromatin. First, coming to DNA methylation, first I am going to read it. Reversible changes, that expression of DNA segment without changing its sequence, involved with aging, carcinogenesis, epigenetics, genomic imprinting, transposable element repression, and X chromosome inactivation. DNA methylated in imprinting. Methylation within gene promoter CpG islands typically represses silences genes transcription. CpG methylation makes DNA mute. Dysregulated DNA methylation is implicated in Fragile X syndrome. Basically, what they are gonna tell over here is methylation mutes DNA. What do you mean by muting of DNA? So, when you have a segment of DNA, you need it to transcribe into mRNA, and this forms the protein by translation. Stopping of this whole process of converting your DNA into RNA and protein is called muting. This is done by methylation. So here the point that is to notice is regulated DNA methylation is associated with the Fragile X syndrome. This is an important point. Here is a mnemonic: methylation makes DNA mid, and yeah, methylation within if the see what do you mean by promoter? If this is the DNA, during replication it unwinds itself, so and it is stabilized by some molecules. We'll see over the next few course. So if this is the promoter sequence, if we mute this promoter sequence, so the transcription does not happen, so the RNA does not form. That's it, mutes. Histone methylation, DNA methylation, histone both mutes the DNA, but in this the DNA is methylated, in this histone is methylated. That's the difference. Usually causes reversible transcriptional suppression, but can also cause activation depending upon location of methylation. It can also cause activation, not only methylation. Not only muting of DNA, but mostly it makes the DNA mute. Lysine and arginine residues of histone can be methylated. We know the lysine and arginine provide positive charge. If you give methylation to it, more positive charge, more binding to DNA, and decreased activation. This is what happens with histone acetylation. And if you methylation, if you go to histone acetylation, removal of histone's positive charge relaxes DNA, increases transcription. Thyroid hormone synthesis is altered by acetylation of thyroid hormone receptor. Histone acetylation makes DNA active. What do you mean by this? If your histone core has positive charge and your DNA has a negative charge, this histone positive charge is provided by large, remember large, lysine and arginine. So if you acetylate, this positive charge will decrease. What will happen if your positive charge decrease? Your positive charge is decreased between your DNA and histone core. So what will happen? There will be no formation of core nucleosome. So obviously there is no transcription. Histone deacetylation, removal of acyl groups, tighten DNA coiling, decreased transcription. Histone deacetylation may be responsible for altered gene expression in Huntington's disease. This is important. Where did we see Fragile X syndrome? We have seen it over here in DNA methylation. Fragile X syndrome. Here it is Huntington's disease, histone deacetylation deactivates DNA. Now we will go to the euchromatin and heterochromatin. First we will go through it. Heterochromatin, condensed appears darker on electron microscopy, sterically inaccessible, thus transcriptionally inactive, increased methylation, decreased acetylation. Heterochromatin, highly condensed, hidden chromatin. Bar bodies, inactive X chromosome may be visible on periphery of nucleus. So euchromatin is less condensed, appears lighter on electron microscope, transcriptionally active, sterically accessible. Truly transcribed euchromatin is expressed. You are going to understand it. So your nucleus has DNA which is condensed around your histone octamer. If there is tight attraction, there is decreased transcription, obviously decreased protein synthesis. So if this is tightly adhered to each other, this is known as heterochromatin, which is inactive form of chromatin. When your cell needs something to synthesize, then you need to form euchromatin. How do you form euchromatin? By acetylation. Because acetylation activates your DNA. So less condensed is more acetylated. Transcriptionally active, sterically accessible. This is euchromatin. So in heterochromatin there is increased methylation. Here there is increased acetylation. So this is how we completed the first page. Yeah, this is an easy one. I'll explain this page and over here the purine salvage deficiencies in the same way. Yeah, nucleotides. So basically nucleoside is formed by base plus ribose sugar, nucleotide by base plus phosphorus, ribose plus phosphate, linked by 3' 5' phosphodiester bond, 5' end of incoming nucleotide base the triphosphate. Purines, AG, 2 rings, pyrimidines, cut 1 ring, pure as gold, cut pyrimidines, thymine, methyl, thymine has methyl, CG bond stronger than AT bond because it has 2 bonds, increase CG content, increase melting temperature of DNA because CG has 3 bonds. So if there are 3 bonds which is more stronger, so you need to melt it more temperature, which means your DNA is more stable. See, here is the mnemonic. Excellent. Mnemonics are came in first aid. Crazy CG bonds are like crazy glue. Then you wait here. Deamination reactions. These are important. If cytosine is deaminated, you get uracil. Adenine is deaminated, you get hypoxanthine. Guanine and deaminated, you get xanthine. 5-methyl cytosine deaminated, thymine. Uracil is found in RNA, we know it. Thymine is found in DNA. Methylation of uracil makes thymine. This is important. Uracil on methylation forms thymine. How to remember this? U met, which means uridine, uracil on methylation forms thymine. So this is a mnemonic over here. Yeah, amino acids are necessary for purine synthesis. These amino acids glycine, aspirin, aspartate, glutamate. This is important. GAG, very defined GAG and GAG reflex. Here this is GAG. See, you have purine, this is pyrimidine. So you have aspartate, you have glycine, you have glutamate, which are nothing but GAG, glutamine. Sorry. Before going to the de novo pyrimidine and purine synthesis, I would like to visit this purine salvage deficiency. Why? Because we have seen already. Your free coming up from the down, your free bases are forming nucleosides. If your nucleosides have phosphate, then it forms nucleotide. This nucleotides forms nucleic acid. So this is the form. This is anabolism. In catabolism, your nucleic acids are converted into nucleotides and then nucleosides and then free bases. So in the process of the degradation, we have some salvage pathways. What do you mean by salvage pathways? These salvage pathways will again convert all your nucleosides, nucleotides into nucleic acids. So here it is, see, we have studied there, we have sugar base either for nucleoside or for nucleotide. So this is the ribose sugar. De novo synthesis, which is synthesis newly, we synthesis again freshly. Your nucleic acids are forming GMP, which is GMP is nothing but nucleotide. Nucleosides guanosine, which form guanine. Guanine is a free base, hypoxanthine is free base, adenine is free base. If the hypoxanthine is forming a nucleoside, it is inosine. If it is forming with phosphate, see, phosphate groups over here. If there are phosphate groups, then it is called nucleotide. If they form nucleic acid, that is nothing but DNA and RNA. So, thing to remember, if you have phosphate, make it as nucleotide. Okay, I am just wiping off all this to get more clear. Yeah, here the important thing is see, adenosine deaminase APRT, adenosine pyrophosphate, ribosal phosphate, GPRT, hypoxanthine guanine ribosal transferase, exoxanthine oxidase, SCID. These many syndromes are associated with your salvage pathway. That's why stressing on this is very important. See, in Leach-Nyhan syndrome, GPRT deficiency. What do you mean by GPRT? GPRT is nothing but which again forms your nucleotides, which is nothing but your salvage pathway. So, if this is deficient, we have Leach-Nyhan syndrome. The classic thing in Leach-Nyhan syndrome is there will be peripheral biting. The child bites his own fingers. And yeah, see, these are adenosine deaminase deficiency, which formed SCID, which is the most common. It is autosomal recessive, and we have another form in SCID. That's not important. IL-2 receptor deficiency, which is X-linked recessive. So these are the two forms, and the X-linked recessive is the most common form. You have cladribine, you have pentosine, which inhibit adenosine deaminase, and you have see, urate oxidase. This has a separate story. Urate oxidase is generally found in birds. What they excrete LN1. So they convert this uric acid by forming this uric oxidase. So scientists have developed a RASP uric acid. This is not found uric acid excretion. Uric acid is not converted to allantoin in our humans. These are found in birds. So this enzyme is prepared pharmacologically and is known as RASP uric acid. Your L-pyranol and febuxostat, these are all nothing but used in gout. Gout is nothing but increased uric acid levels. Okay. Adenosine deaminase is required for degradation of adenosine and deoxyadenosine. So if there is decreased adenosine deaminase, your AMP is not forming adenosine, or so when this is not formed, there is increased ATP and decreased nucleotide reactase activity, decreased DNA precursors in cell, decreased lymphocytes. One of the major causes of C, autosomal recessive. You have also X-linked recessive. This is not important here. Leicht-Nyhan syndrome, defective purine salvage pathway, absent HGPR. See, this degradation pathway defect. This is salvage pathway defect. Decreased CGMP, decreased IMP, because you are not reforming all these. So there will be compensatory increase in purine synthesis, increased PRPP amino transferase activity, excess uric acid production, X-linked residue. See, self-mutilation is nothing but biting your peripheries, fingertips, hyperuricemia, red orange, and sodium urate crystals in diaper. This is important. Sodium urate crystals in diaper. Gout dystonia, macrocytosis, HGP at hyperuricemia, gout, pistoff, which is aggressive and self-mutilation, red orange crystals in urine, tense muscle, treatment is allopinal febuxostat, which increases the excretion of, decrease the formation of uric acid. This is important. This decrease the formation of uric acid, whereas urate oxidase will increase the excretion of uric acid. So this increase excretion, these are decreased in the formation.